Okay, do you hear me well, please, there? Hello?
Yes.
Okay. All right, okay, that's fine. All right, let's try to connect here, first of all, to see how it's going to work out. Okay. Okay. Well, I guess from last lecture, I started with you with this phylum, okay, basically just to make sure that just, okay. Of course, we are dealing with the just general phylum, what you call Dicaryomycota, okay. And of course, we are dealing with a second group. I'm just directly going to go. We are dealing basically now with the class Basidiomycetes. Okay. And already I started with you last lecture about the general characteristic of this group, okay? And we talk a little bit about some other criteria, for example, like the presence of clump connection. And the last thing I was talking to you basically with regard what you call to the pores, okay, which already I mentioned that for you last lecture, okay? Just to connection here, simply to mention this point, okay? That the septa, because as you know, the septa, which is between the cell, already I mentioned that, but at least to repeat it again here, that the septa of the most of Basidiomycetes are swollen, okay, and centrally perforated, okay? Now, as I mentioned however, most of the Basidiomycetes, okay, that the both group, I'm going to mention the whole of Basidiomycete and Phragmomycete, okay, the septa are, because in Basidiomycete you have three groups. We have based on the, what you call, on the basidium, we have the holobasidium, and we have the phragmobasidium, and we have the teleomycetes. So the two group basically, the holobasidium and the phragmobasidium, that their cell, the septa is swollen, okay, and swollen basically, and the central pore basically have a central beric-shaped structure, and this is what you call the dolipore. Okay, we call the dolipore. Already I mentioned that to you here. Okay, now and basically and covered on both sides by a cap membrane called parenchystomes. Okay, covered by called parenchystomes. Already I mentioned that to you here. Okay, then I mentioned in the case of teleomycete, the septal pore is simpler and often blocked by polywheel occlusion, called by polywheel occlusion. Did you follow that? I'm asking you, or not?
Yes, we follow.
Okay, so this already I mentioned to you last lecture. Now I'm going to continue after that. Okay, now. Okay. Then I'm going to start here now basically the type of basidia. Subtitle here, types of basidia, which make it more easier, and of course in Basidiomycetes. Of course, we have three types here. We have the holobasidium, okay, and also we have the phragmobasidium, and the third one we have is known as teleobasidium. Okay. Now, this one is here, basically just, I'm going to put the note, but let me first explain it for you. Okay. Now, as you can see, the whole of basidium, this one here, okay, irrespective of the shape, because we mentioned, of course, they have a basidium. So that means the basidium cell is one cell. Okay, and that's the whole of basidium. Irrespective what shape it has, as long as there is one cell, in that case known as what you call the whole of basidium, or the other, in which basically the basidium is fragmented, as you can see here, 1, 2, 3, or and that is you end up with 3 cell or 4 cell, and this is known as phragmobasidium. The Tiliomyces, which is occur only in one group, basically in the rust plant, or in the rust fungi. This one here, the rust fungi, of course, these are the most parasitic. At the end, we're going to talk about it, in which basically they, what you call the wheat and other, they when being infected, they produce what you call a rust color on the leaf, and that's called them the rust fungi. So basically, these fungi under this group known as Tiliomyces, in which they have 2 cells like this, and then they will make a germ tube like this, and then the basidium occur on the top like this. So basically, these are the 3 types, and based on this 3 type of basidia, we are going to have the different group which they show the one of the type of these basidia. So basically, these are not. So the whole of basidium simply is going to mention to you here, okay, that it is one cell structure. Simply to mention is one cell structure, okay, of varying shape. Okay. Then the other one, which is, okay, let's see how this. Okay. And then, of course, we have the second one, which is the phragmobasidium, okay, which is simply going to mention here that a septate basidium. Okay. A septate basidium with one or more a transverse or vertical septa, with one or more a transverse or vertical septa. That means, okay, when you have it like this one here, okay, that means when you have like this one, this is called transverse septa, or in some group it could be vertical septa. But in the end, you end up with many cells that the basidia is not one cell. Okay, then we go to the last one here, the Tiliomyces. I'm going to discuss it to write the note here. Okay. So as I mentioned to you, which consists generally which consists of a generally thick-walled we call them thick-walled resting spore. Okay, resting spore. Okay, and affinate germ tube. Sorry. Okay, which issues from it okay, which is from it, okay, and beers, and okay, the basidiospore. So as I mentioned to you that basically I'm going to that you have a finite with a not indefinite, a finite, that was a finite German tube or like a mycelium, and from it depend what you call produce these basidia or basidiospore. Okay, so basically these are the three types of basidia which are produced by the basidiomycetes, and of course when we discuss the group, depend on this type, each type on this type of the basidia. Okay. Then after this, I'm going to go and discuss with you basically what you call the different group. I'm going to start first because the one in which basically their basidia is one cell, which is known as holobasidium, and this group we call them holobasidiomycete. The first group is going to be the holobasidiomycetes. Okay, known as the holobasidiomycetes. Now, in this group, basically, we're going to have, we're going to have two. Basically, in this one, basically, we include almost of the mushroom, and basically, all the mushroom, basically, including this one here. Now, this one, as you can see, okay, just give me a second to explain this one, okay? We have different type, as you can see now here. Did you see that? We have different type. Now, even if you look at the mushroom, whether it is the foliage or the Gill mushroom, the one you're already familiar with, basically what happened here, under the cap here, basically you have a line and you have all the basidia in one line. You follow that? And that's we call them hymenium. Okay? So basically in which they produce the basidia in one line, basically they make it, like if we take the mushroom basically and you try to look at it, it looks like this one here, okay? So basically what you have, we follow because the mushroom is nothing more, as I mentioned to you, a packed fungi. Look, okay? So if you take the mushroom and you tease it, basically it's a collection of fungi, but they very packed, look like a tissue. Okay, so basically what happened here, as already explained, when you have all the basidia in one layer, like this one, which is known as a fertile layer, this is then called hymenium layer. Okay, so basically what happened here, all this holobasidium in which, okay, they produce what you call their basidia, and this known as hymenobasidiomycete. Okay, and also we have other type of different group in which basically the basidia is not in a hymenium layer, okay? Like for example, like the puffballs, okay? Like this one here, okay? Like this one. Have you seen the puffball? Like puffballs or earth fungi or the bird nest fungi, in which case, of course, the puffball, if you take it and you make a cross section, you find there is many chambers, and in each chamber you have thousands of basidia. So basically, the basidia are not lying in a hymenium layer. So basically, what happened here, going to group this, the whole of basidiomycete in two groups. Those one in which they form the basidia in hymenium layer, or those in which the basidia are not in a layer or hymenium layer. So this is what we basically want to talk about. Okay. In general, of course, as the whole basidiomycete, the main characteristic or the chief characteristic of this group, okay, of this group here, which means the whole basidiomycetes, okay, of this group of fungi. Okay. Are you okay there? I don't know. Oh, sorry about that. Seems I pushed something here. No. Okay. It was basically what you call that. Okay. Is there. Okay. Typically. Okay. Non-septate. Okay. Basidium. In which basically known as the holobasidium, which is already explained to you. Basidium, which usually, okay, bears. Of course, don't forget that this is the holobasidium or the cell in which in it occur what you call the meiosis, and then of course producing after meiosis the four basidiospore. Okay, which usually bears four basidium spores. Okay, four basidiospores. All stigmata. So basically, what is their characteristic here? Okay, to mention. Now, to mention, of course, the other point, there are, okay, two, okay, natural groups okay, within the holobasidiomycetes. Now, this group, okay, we call one of them hymenomycetes. Okay. Which is simply that this group, as a matter of fact, they bear, okay, their basidia. Okay, they bear their basidia in a well-defined hymenium layer. Okay, hymenium layer. And of course, all the basilospore are exposed, okay, which becomes exposed. Okay, exposed. While the basilospores okay, are still young. Okay, and undeveloped and developed. So this is one group, as I already mentioned to you, in which basically all the basidia, okay, in one hymenium layer. Just give me a second here. Then, of course, we have the second group, okay, in which known as Gastromyces. Okay, of course, this one basically, as I mentioned to you, in which the bacilli are never being what you call shown or an hymenium layer. So simply to mention, they never expose their, okay, simply to mention, they never, okay, expose their basidia, okay, in a hymenial layer. Okay. And that's why basically what happened here, because when the basidia is exposed in a hymenial layer, of course, that means the bacillospores are able, what you call, to disperse themselves. In this case, of course, only when the fruiting body as a whole is going to decay or to break, and that is basically when the spores are released. Okay, to mention to you here, okay, that means in which, well, okay, and I will mention that in some Gastromyces, this is G here, continue here, some of these Gastromyces, okay, the spores, okay, remain within, of course, now the fruiting body we call basidiocarp, okay? As before in the ascomycete, we used to call ascocarp or ascoma. Here we call basidiocarp, is the fruiting body, okay? Within the basidiocarp, okay? And of course, and are liberated means and are. When the spore are liberated, of course, only when that fruiting body is broken or decayed. Are liberated only upon, of course, breaking or we call decaying or weathering of the fruiting body, or of the fruiting body. Okay? Now I'm going to talk the example for each one here. Okay, basically, as I mentioned, that most of the now I'm going to start first, of course. We're going to start first with the Hymenomycetes. Okay. And simply to mention here the familiar example. Of this group. Are, for example, mushrooms. Okay. Known as the boliots. Okay. The other one, which is also known this one here, what you call the bracket fungi. And also we have the corals or the coral fungi and the tooth fungi. I'm going to explain some example of this one here. Okay. We're going to start the first group here, which is okay. We call basically what you call the gill mushroom. Okay, and I'm going to give you this example on the gill mushroom called Agricus campestris. Okay. This is basically the gill mushroom here, which is like this one here. Okay, I think you have seen this one here. Basically, this one here, basically, if you look at the back of the mushroom, it's going to look like this radiating. But really, if you examine all these, we're going to find it's all basidia and basidiospore, or this one here. Okay, so basically, these are the gill mushroom here. Okay, so first of all, we're going to talk about what you call the structure. Okay, and actually, even the term mycology, as a matter of fact, which we learned, is derived, as a matter of fact, from the mushroom, because mushroom in the Greek, they used to call it Machaeus, and from that terminology came the term mycology, okay? Just for your information here. Okay, now, talk about here the general morphology or structure of gill mushroom. We call it gill mushroom because, as I already showed you, because of these gills at the back here, okay? So basically, these are the structure we're going to know, okay, of the mushroom here. Do you follow that? Okay. Because I see you can have the cap, you have the cap here, which is called Peleus. And of course, here, as you can see, the gills, okay? And of course, here we have the stalk. And basically, sometimes you can see a structure like this, a small membrane, which is basically that from the V was covering the whole mushroom. Because the mushroom basically is small like this, which we call the bolts under the soil, and later when open, then that membrane is going to tear off, okay? And then, of course, we have in the lower part here, which looks like a cup, we call the vulva. So basically, I'm going to talk about that structure here. We're going to describe it, okay? Now, to mention you that a mushroom, okay, consists of a stem, the one I showed you, or we call steep. Okay, and of course the steep or that stalk supporting supporting a cap. And the cap we call Peleus. Between two brackets known as Peleus. Okay. And that stem, as a matter of fact, as I showed you here, will rise from that cup. That is the stalk or the stipper. And of course, this is the cup under the soil, which is known as the vulva, to mention this point here. So basically, as I mentioned, a mushroom consists of a stem, which is a steep, okay, supporting a cap, okay, and sometimes rising from a basal cup. And that cup basically is known as the vulva. Okay. Now, of course, as you can see, the gills, on the left, they are the lamella, known as the lamella or lamellae. Okay. Which is basically what they are. They are plate-like structure. Okay. They are plate-like structures radiating from the edge inward. Okay. Of course, toward the stem. Okay. Of course, they are under the pubis. Which is basically the cap. Okay. And basically, of course, they are all covered. All these gill are all what you call all the gill are covered. They are all covered. Okay. With basidia. Okay. With basidia. Okay. On both their surfaces. Okay. That means if you try to show it this way here. Okay. That means if this is the lamella here, so basically they have the basidia on this surface and this surface. Do you follow that? So in both the surfaces of this gel here, you have the basidium with the basidiospore. And simply, of course, sometimes if you take a mature mushroom, you cut the head, and if you bring it on a white paper and you tap it like this, you can see all the spores going to fall on the paper, forming the same form of radiation. Okay, you can see that. All right, to mention this point here. Now, as I mentioned to you, that means with the endobacterias are covered with basidia on both their surface, so to mention to you here. Okay, but also don't forget, of course, that basidia which inside occur the meiosis and later on produce what you call the basidiospore. Okay. Now, and to mention this point here, their surfaces, and to emphasize this, that each basidium that each basidium generally producing 4 spores four spores, which are the basidiospores, and usually that occur after karyogamy. Okay, are you following that? Okay, after karyogamy, all right, and meiosis. Okay. Please don't hesitate to notify me if I basically go down and you don't see the writing. Okay, just let me know. Okay, and karyogamy and meiosis have occurred. All right, now, and sometimes, of course, the other point to mention to you here, okay, that Also, there is on the stem, and there is, okay, a ring, which is known as annulus. I'm going to show it to you here, known as annulus. Okay, around the stem, or the steep around the stem, below the edge of the perius. Okay, talking about this one here. I think I can have a picture. Yeah, this one here. That, for example, as you can see, this is shows you here in the figure. Give me a second. Talking about this one here. This is called here the ring, but in reality, it is also known as the annulus. Okay, and that is the ring, as I told you, is the remaining of the universal veil, we call it. Okay, and sometimes it shows like this. Did you see this one here? Shows like this. Okay. As I mentioned, called known as the annuals around the stem under the perius. And as I mentioned to you, simply to me that this represent this represent the remnants of the velium. Okay, so you have to know all this terminology, okay, of the velium, okay, and basically this velium, as a matter of fact, okay, which covers, okay, which covers the gills, okay, before the spores mature. Sometimes if you take even if you go to the market and you take some of these mushroom, the edible one, and really if it's very young, basically if you look under the cap, you can see it's still, it is covered with that velium, okay? The only time when the spore mature, then that cover is going to release, okay? And that's a mention that represent which cover the gills before the spores basically mature. Now, the other point we're going to talk about is this one here. I think you have seen some kind, if you walk in the bush or something like that, you find basically when you have the cap like this, okay, and then you can see, especially what you call the ammonia mushroom. Sometimes if you walk in the bush, you can see it's red, called ammonia, but you don't touch that. It's very poisonous, called ammonia mushroom, okay? Okay, and you see on the top, there is a lot of cover like this dots. Sometimes gray or whitish color. This is, of course, the remaining of the also of a membrane or entire view. To mention to you here now. Are you following what I'm talking to you here? Okay. Now, to mention this point, the peleus may also, okay, be covered, okay, with called scale-like membranes. Okay, and this one basically these, as a matter of fact, these are the remnants okay, of what you call of the universal veal, of the universal veal, okay, that usually used to cover the whole mushroom when it was a small, and usually when it is a small, like a button, just to mention to you here, okay? Membrane, the remnants of the universal veal, okay? Which is basically, what is the veal? Which is a membrane, okay, that covers, okay, basically the entire, just to cover the entire, okay, the whole fruiting we call the sporophore is the whole mushroom, okay, in the younger stage, in the young stage. And this younger stage known as the button stage. Known as the button, okay, which is this one here. This one here, okay. So basically, when, as you can see, the stage is now when mature, okay, gradually, of course, that when it tears out, then it's going to be as a remnant on the surface of the cap here. Did you see that? So by this way, basically, you have all the structure of the mushroom, and basically in which you have to know all this structure there, okay? Now, as I mentioned, the other thing I would like to add to this one here really is simply to mention that for you to know that the mushroom body as a whole, the one you see, the mushroom body is no more than a compact hyphae together. Just to make sure it's no more than a compact hyphae, that all the structure of the mushroom you see is no more than a compact hyphae. The other point I'm going to mention to you here, okay, that species I told you, which is Agrestis campestris, that is basically, of course, is the only species which is basically was able to be cultivated. Because there is a lot of mushroom cannot be cultivated because some of them, they develop a mycorrhizae, and sometimes they, though it looks in the ground, but they cannot tell where, okay, with which species, okay? So basically that is the only two cultivated species so far is the what you call the white mushroom you see in the supermarket, which belong to this species, Agrestis campestris, and the other one, maybe you have seen it in them also the supermarket, called the Shiitake mushroom. You know that? Which look like brownish in color. So I'm going to talk about these two things here. Okay, just mention something here. Okay, just to mention, of course, that a great industry yeah, a great industry has been developed okay, on cultivation of this species I mentioned to you of what you call Agriculus campestris, which is commonly also we call it because gill mushroom could apply to many species. To Agriculus campestris Okay. So basically, this is to mention basically the only mushroom, okay, has been cultivated of course for consumption, I guess, okay, on a large scale, though I mentioned basically in North America, but also I guess in Europe and other places they can also cultivate it. Okay. The other one, of course, is the Shiitake mushroom. We're going to mention about it. Okay. Also the cultivation. Okay. Of the Shiitake mushroom. And this basically, this mushroom is more consumed in the Orient, like in China, in Japan, and in that place, okay, or Taiwan, okay, mushroom, okay, which is basically that shiitake mushroom. As a matter of fact, belong to the species called Cortinellus abricanus. Going to mention to you here, called, okay, Cortinellus, okay. Abricanus. And as I mentioned to you, has as a matter of fact, has been expanding in the Orient. Okay, like for example, Japan and Taiwan. Okay, mainly. This is again to mention just another example. I don't know. I think some of you sometimes, if you, I don't know if you have seen this one here. Sometime you wake up in the morning in some yard, or you find some mushroom. They are forming a ring like this. Have you come across something like that? Okay. These are special mushrooms known as fairy ring mushrooms. Okay, I'm going to mention it to you. Sometime also you can see, okay, in some garden or lawns, okay. It depends. Just to mention this point here, okay. The example I'm going to mention to you here, I'm going to use this one like this, okay. Okay. Another example known as the fairy ring mushroom. Okay. Also, it is another species, of course, of the gill mushroom here. Okay. And the fairy ring mushroom, the Latin name for it is known as Maracimas. Okay. Orieris. Another example of this gill mushroom called. The other one is this one here, if you want to put it down. This one here. Another example called Lipotia species. Also another example of the gill mushroom. Okay. Then I'm going to move. Basically, what I'm doing is this one here, just for your information, so that something. I'm trying to give you this example here, okay? Basically, as I said, I give you this is the Gill mushroom. Then I'm going to talk about the shell fungi here, and then talk about the bullets here, okay? I'm going to show to you what I showed the color one, okay? Still, this is one example we talk about basically under the Hymenomycete. The other example I'm going to talk about here now. Okay, just paper around here. Okay. The other example, or put example number 2, whatever you want to put down. Okay. Known as the bracket fungi. Okay, or sometimes we call it the Polyporus. I think most of you have seen this one, for example. If you walk in the wood, and especially if you see some of these old trees or even dead tree, you find there is a fungus basically is attached to the, what you call, like this to the tree. Like this one here. This is known as, we call as a bracket fungi, or we call it as a polyporous. Because what happened here, if you take the thing, I don't know if they showed you in the picture in the lab, if you look underneath it, you find it has many pores, okay? So many pores, and each of these pores is the bacillial spore are lining around it. And that's, we call it what you call the polyporous, okay?
Sorry, just a random question.
Yes.
Going back to the fairy rings, why do the fairy rings grow in circles?
Because what happened here, because the fungi, basically what happened here is see what you call the hyphae, because the fungus is growing from the hyphae under the soil, okay? And if they have a symbiosis, and that's why they go around like this, one to another like this, okay? That's the way they grow, okay?
So you're not looking at just like, when you look at a fairy ring, you're not just looking at one organism. You're looking at a bunch of individual fungi organisms that are symbiosis together.
Yeah.
Okay.
Go together, okay? Yeah, because sometimes it's well known that basically most of the mushroom, which they grow, of course, you can see it in the forest, okay? All of them, they grow in one way or another in symbiosis or in mycorrhizae with other species. Tree, shrub, and so on. You get my point of view here, okay? Because really, I didn't have much time to discuss with you the life cycle, which is a little bit too complicated, okay? Of the mushroom, though it seems very simple, okay? But it's very complicated life cycle it has, okay? When it do, especially the sexual reproduction. And now we're going to talk about the bracket fungi I mentioned, or we call it the polyporous fungi, okay? But the species, the example we have here, called Polyporus sulphureus. And of course, they play a big role, this fungus, as a matter of fact, in decaying the wood or the lumber, okay? And, okay, so I'm going to show you. The dry one here, I have the picture of the, okay. Sometimes it looks like this one here. Okay, like this one here, sometimes look like, especially of course they dry out when they're becoming old, but when they are still young, they are very soft and really according to supposedly be edible, but I don't advise anybody to eat from mushroom unless you have some experience or somebody with experience, otherwise you'll end up with a poison. Okay, now to mention this point here. Now this is the polysulfuronous. Okay, now to mention this one here, basically in the polyporous, because I mentioned it has hole underneath it in the polyporous. As a common, okay, the basidia, okay, line, okay, the inner surface of the pore, line, the inner surface of the pore. Like basically, like the bullets, I'm going to just give it, okay, but the bullet, of course, they have a thick head and very fleshy, but the bullet also, they will form pore. They will show you what we are talking about here, okay? Yeah, this one here. If you focus in this one here, this is the gill mushroom, the same thing. You see, this is the bullet. If you focus here, you can see there are all pores. Okay, so the same thing with the bracket fungi. And inside these pores, underneath it, you find basically all the basidia inside. So that basically that's what you are talking about. But this is of course different. But I didn't have what you call a back picture of the bracket fungi, and that's why I tried to show you this one. So as I mentioned to you, in the pores, the basidia line the inner surface of the pores, okay, or tubes, okay, beneath the cap. Okay. To mention this, of course, the fruiting body, to mention here that the fruiting body, okay, may resemble, okay, may resemble, of course, crusts, because it's, especially when it dries very hard, okay, crusts, okay, or sometimes like shells, okay. Now, most of these one, of course, as you can see, they are the bracket fungi, they are sitting on the, so basically they don't have any stalk, maybe some of them there, but in general, that's what they are, okay? And to mention to you that these basidiocarp, in the fruiting body, these basidiocarp may or may not may or may not have a stem. Is it okay? Sometimes they are sessile, okay, on the stem. Of course, something to mention, of course, and sometimes also we call these bracket fungi because of their red, sometimes we call them also sulfur fungi, okay, or sulfur mushroom, okay? Now, of course, some of these they cause quite a disease in the lumber, okay, because that's what they grow. Just to mention to you, many species, this is many species, okay, many species of Polyporus, okay, cause disease, okay, of forest, okay, and shade trees, of forest and shade trees, okay. And as I mentioned to you here, also others are important. Others are important, Okay, because what you call because they attack and destroy lumber. Okay, and destroy lumber. Because that's what they. That means what it is. That means they have, of course, very simply. That means because the wood, as a matter of fact, because the wood, as a matter of fact, is the xylem. Okay, and one of the compound in the xylem, of course, you have, of course, plus the cell wall as the cellulose, and also you have the lignin. So in other words, that means these fungi are produce the enzyme which can degrade what you call the lignin and also the cellulose. Okay. Now, going to move to another example here, which is also as of these the Hymenomycetes, okay, which is known as the bullions. Another example here, going to mention to you. Still, do you see this? Okay. Bullions. This is the name of the species. Species here, and this is the one I showed to you, I guess. Yeah. In which basically it has a very thick head. Okay, a very fleshy head. Okay, just. Okay, so basically these are the bullions. Okay, but of course, what is okay? It has a very thick, fleshy head. Okay, but of course, also underneath the cat is all pores like this one here. Let's talk about this one here. Okay. Now, to mention that this group of fungi. Okay, this group of fungi, as a matter of fact, here, okay, also are mushroom-like. Called mushroom-like because they're similar to mushroom you are used to. Are mushroom-like. Okay, in their general aspects. Okay. Okay, in their general aspect, and to mention this point here. Now, of course, their characteristic, what they have. Now, the thick, they have the thick, okay, fleshy cap, okay, beers, okay, on the under, okay, on the underside, a layer, as I showed to you, a layer of tubes, okay, or pores, a layer of tubes or pores in which the bacteria are born. Of course, in which the bacteria, and of course the bacteriospore, in which the bacteria and bacteriospores are born. Okay? You follow that? Okay, you can add to that basically that, you know, the same thing as I mentioned to you, that many species of bolete also what you call, they form mycorrhizae with many tree species, okay? Of course, some of them are edible and some are poisonous to me. Now I'm going to, the other example I'm going to give it to you, I don't know if you have seen this one here, called Polyporus mexicana, or sometimes we call them the hallucinogenic mushroom. The other example I'm going to give you here was this one here, okay? Called Sirio. That is the name of the species. Okay, Mexicana. Have you seen this one or no? No? And this is basically known as the hallucinogenic mushroom. Okay, sorry, the hallucinogenic mushrooms. Okay? Because they have a compound, just, okay, they look like this one here. This one. You see it? Called the Polyporus mexicana. Okay? And of course, I'm going to mention a few lines on this one. It is basically some tribe, they use it. They go to for about a few hours, but of course, later on, they, what you call it, mention that. And of course, there are two scientists when they try to discover the reason for that. Okay, just to give me a second here. Okay, now. I mentioned the scientist. There are two scientists here. They went to that, I'm going to mention here. The scientist, we have what you call. One of them is the French mycologist. Just called Roger Heim. Okay. And his American sponsor, they visited, of course, which is named that R.G. Wilson. Okay. They visited Mexico. Okay. And of course, and took part because they saw that the people, as a matter of fact, took part. Okay. We called in the sacred mushroom. It used to use by some of the tribe. Okay, used. Okay. Used by some tribes there. Okay. Like they do used to as some ritual or something like that. Okay. And he described that mushroom basically as this, okay? And later on, okay, and later on, basically what you call the Swiss chemist, which his name was Hoffman. Basically, he managed to isolate that compound in which they used to cause what you call the hallucination. And he found they are but there are two compounds, and of course, he named them, of course, based on the name of the species itself. Okay, isolated, what you call the hallucinogenic principles. Principles of compound, okay, and he named them, okay, and I've just put here, and name basically on the name of the species. One, because there are two compounds. One is known as psilocybin, because from the name of the species itself. So one named as psilocybin, and the other named as psilocybin, and the other one known as psilocybin. So he named the two compounds which can cause that hallucination, but of course it takes about 2 to 3 hours, and then of course the compound will lose its effect, okay, or effect. Are you okay? All right. Well, it seems the time is, yeah, still I have some time here, I guess. Okay. Then we're going to move basically to the other group, which is basically the, okay. The other fungi is known, I'm sorry, I was losing my voice here for some reason or how. Okay. And the other example I'm going to give you, okay, known as the coral fungi. Okay, before the time will pass on this, will shut off here, okay? So the coral fungi, they look like this one here. Did you see this one? Okay, but of course they are very beautiful. If they look to the color, they look like this one here. Okay, sometimes they look like this one, or they look like this one. But as you can see, they have ridges. It goes down like this one. That's what basically I am going to describe it for you. Okay, known as the coral fungi. But as a matter of fact, they belong, or sometimes we call them the funnel fungi. And also it's known as the funnel fungi or funnel shaped. And of course, all these basically and belong to this family called Calvariaceae. Okay, and the example of the genus is known as Calvaria. Okay. Yeah, these two, I'm going to put one line. Now, these fungi, before that, do you see it now? These fungi, okay, with shallow or deep ridges running, okay, from the underside okay, of the funnel down into the stem. You follow that? Because after this, I'm going to stop my lecture. Don't like to cut me off. This one here. Okay. These fungi, of course, they call them coral because they show the same color. Okay. The same color, basically like the coral fungi. We're going to talk about next lecture. And of course, they show a very nice, what you call. The other one, I'm going to talk about it also with these. They are all with the type of the Hymenomycete, okay? Because they form the basidia layer. The other one is known as what you call the tooth fungi, like this one here. Okay, you see these cool tooth fungi. Also, they're going to explain this one, but also they are really, when they do, they are very beautiful. Okay, these are the tooth fungi. They develop something like that, and sometimes very white. Okay, so these are the one I talk about it before I will come. Then these what you call the coral fungi or the tooth fungi is going to be the last of the Hymenomycete, and then I'm going to start with the Gastromyces. I'm going to give you, of course, the puffball and the earth star and the bird nest fungi. Okay, these are the one, and then going to move to the other one. Is that clear? Do you have any questions? No? Okay.
Oh yeah, Professor. I just had a question about the essay that's due at the end of the term.
Yes.
So is it due like on the day of our final exam, or do we have to like give it in before that, and the essay in before that?
Is the final exam is out now?
I'm pretty sure it is because they sent out.
Yeah, they sent me the email because really I didn't open it. I will let you know. Usually you got my point of view, but normally what I said, as long as you send it to me before the final exam, okay?
Okay. And we're not going to have like a second midterm, right? It's just straight the final exam and then the essay.
Say that again.
We're just going to have like the final exam and then the essay,